Hello, my name is Meow, and I'm doing a live stream today talking about metabolism. My background is that I'm a molecular biologist. I studied genetics at UNSW, and since then I've been very interested in life, especially at a subcellular level. Um, I'm going to be talking about metabolism without thinking about the actual organism it comes from. We'll talk a bit about human stuff, but I'm going to be talking about metabolism in general. Like, what does it actually do? What is it? This actually comes from a Greek word. So metabolism is comprised of two parts. It's catabolism and anabolism. So anabolic, like anabolic steroids, makes to grow more. So anabolism is the creation of new molecules, and catabolism, which is when we eat food and we break it down and turn it into energy, is kata, to bring it down or down. So the combination of anabolism and catabolism is metabolism. So this is how we break down and create molecules inside our body. And the same processes happen inside bacteria, plants, and fungi. So let's just have a look at what this actually looks like. If this works, here we go. Okay, so I've got a few diagrams I'm just going to go through. Basically, the two main things we're going to be focusing on is carbon metabolism and nitrogen metabolism. So carbon metabolism is a process that happens outside of cells, and it's a process that happens inside of cells. Same thing with nitrogen, and the same thing with fats. But carbon and nitrogen are probably the most important. So in the beginning, all of these elements started as atmospheric gases, and then when life began, it started to use these atoms and molecules inside its own processes. So when we think about life, it's basically a contained system that exists in disequilibrium with the environment. Okay, so there's more things to life, but we can think about it as it's not in balance with the outside. It's got different levels of things than outside, and all it's doing is cycling nutrients. So the obvious one here is the carbon cycle, which is carbon dioxide being turned into sugars, and then those sugars being burned for energy, and then releasing that energy back as carbon dioxide. So a human might breathe in carbon dioxide. It will breathe in oxygen, and it will eat glucose, sugars, and then those will be broken down, the sugars, and the oxygen will be released as carbon dioxide. And a plant will do the opposite. It will breathe in carbon dioxide, create sugar, and release oxygen. So that's a cycle that happens inside us, it happens inside plants, and combined, it happens in the environment. Okay, so let's just have a quick look at the nitrogen cycle. Today we're going to be focusing mainly on the carbon side of things though. So in the nitrogen cycle, there's atmospheric nitrogen. In the air, it's about 70% of our air. Now this gets turned into ammonia by bacteria. They use it in their energy production, and then that ammonia gets taken up by plants and then by animals. And why do we need carbon and nitrogen in the first place? This is probably the most important thing. So every living thing needs three things. It needs, number one, a carbon source. Number two, an energy source. Number three, a nitrogen source. Why? Carbon is what builds most of our skeleton. Everything from fats, which is what all cells are made from, that's what separates us from the environment, through to different things inside of us, all of the different structures and things we're built from, are all made from either carbon or protein. So I'll just bring this across, and then so nitrogen is the proteins, and the energy source is a little bit trickier. So I'll just bring this up. Basically, this is a very high level overview of what you need to be alive. Nitrogen is not included, but let's just have a look. So you can either get your energy from light, or you can get it from chemicals. When we think about chemicals, we're just going to, as a general rule, say you can either be a plant that turns light into sugar, or you can be an organism that eats plants. or you eat an organism that eats plants. But basically, it all starts with the sun. Once you have the photosynthesis and the carbon dioxide, you can make sugar, and then you can either make more of that or you can eat that. Okay, we'll ignore the middle bit. We'll come back to that in a later lesson. And then the carbon source. So plants get their energy source and their carbon source from different places. So their carbon source comes from the atmosphere and their energy source comes from the sun. And then all animals are going to eat it. So the carbon source and the energy source for animals is the same. It's also the same for fungi. So basically you can either make it, which is autotroph, or you can eat something else, which is a heterotroph. We're not going to use these words as much as possible, so don't worry about that. Okay, so that's basically covered. I'm just going to go through this quickly. Basically, another thing to note is that the nitrogen cycles and carbon cycles actually interact with each other, especially when we're talking about things like proteins that are made up of carbon and nitrogen. Okay, we don't need to worry about that too much at the moment though. Okay, so now we've talked about what are cells, how do they use energy, what are they even building, right? So this is a very simple, this way, bacterial cell. Okay, so what's going on inside this bacterial cell? I picked the absolute simplest one. Basically, what a cell wants to do is make more of itself. Not only that, it also wants to cycle nutrients while it does it, and it wants to be in disequilibrium with the environment. Now let's just have a think for a moment. What means that a cell is alive, like a bacterium? And why does that make fire or crystals not alive? So fire is in disequilibrium with the environment in a sense, right? It's a process that's happening, that there's either fire or no fire. So we'd say that's kind of in disequilibrium. The general state of the environment is no fire. It's cycling nutrients, you know, it's oxidizing carbon, which is something that we're going to be talking a lot about during this lecture. And it can reproduce. Little fires can become other fires. It can split in half and make two fires. But there's two key components which aren't there. So one of them is that fire isn't separated from the environment. It is the environment. There's no barrier between fire and not fire. It's a smooth continuum. We don't have that. All living cells have a barrier. In a bacterium, it's called a cell wall. We also have membranes which function kind of like a cell wall made of fats that separate us from the environment. And the other thing is fire doesn't have DNA. So it can't evolve the same way that cells can evolve. And this is important. This is actually what defines life, is this ability to evolve. Okay, so let's have a look at this. We've got something that separates us from the environment. In bacteria, it's called a cell wall. In a human, or most, basically plants also have a cell wall, but everything else just has a membrane, which is very similar to this. We're not going to worry too much about it. Something that separates us from the environment. It has DNA. This is very important. This is the instructions to make everything inside the cell. We have, on a bacteria, it's circular. We don't need to worry too much about that. There's a liquid inside, and all cells have liquid inside. Once upon a time would have come from the ocean, but when it drinks, it will divide, and then that liquid will stay inside, and it will continue from cell to cell to cell. But probably the most important thing is these orange bits here. These are ribosomes. These are pretty much the only thing you need other than separation from the environment, a bunch of enzymes, which are basically little machines that are doing jobs that are encoded for on the DNA, and then ribosomes. So ribosomes turn genetic information into proteins. So basically the DNA is just an instruction manual of genetic information that can be turned into protein. Why do we need proteins? Proteins and carbon make up everything that the cell is made of. And there's no exceptions for that. Like amino acids are made of nitrogen. Amino acids are knitted together to make proteins, and these do jobs. The carbon parts don't really do jobs. Okay, so that's basically where we are. All of metabolism is basically around, you might have heard them called macros before, or macronutrients. And these are your fats, these are your nitrogens, your proteins, and your carbons, which is your carbohydrates or your carbs. So these are the three big elements of metabolism. Today we're going to cover, so this is a map of all of metabolism for humans. Some of this is shared by bacteria, some of it's shared by plants, and a lot of this is actually shared by fungi. So a yeast and a human eat kind of the same. Now, it's very overwhelming when you first look at this chart to think about how this all fits together. But basically, we can break it down into some broad things. So what we're actually looking at is pathways that different molecules can take. Now, basically, this red bit here, that first bit, that bit there, is carbohydrate metabolism. That's how we break down things like sugar and bread and turn it into energy. This is shared by, as far as I know, almost every living thing uses this. It's the way that plants make energy, it's the way bacteria make energy, it's the way cancer cells make energy, even when we starve them in other ways. The blue section here is nitrogen metabolism. It also talks about how we make different amino acids. It looks quite complicated, but basically that's things like ketone body processing and stuff like that's going to be there. And then this section over here, that's a light green, is going to be for lipid metabolism. And remember, the word metabolism is meta, so it's both building and destroying. So in lipid metabolism, it will talk about things like how do we use fat to make energy, but also how do we build fats from other things. In the blue section, we're going to be talking about how do we break down proteins, and then how do we make new ones, and how do we recombine them in these types of ways. And then glucose is how do we break down glucose, but also how do we make glucose. So we generally think about glucose as coming from things like plants, but humans can also make glucose through a process called gluconeogenesis. Now, this section here is something that humans have, but bacteria don't. This is oxidative phosphorylation. So this is how we use oxygen to get even more energy out of glucose. Okay, so that's a basic overview of that entire of the entire process of metabolism. What we might do is dive a bit into the details of it now. Now, I've got the live chat open on YouTube and on Facebook. If you have any questions, or if I say something and you don't actually know what that is, or you want me to go back and explain something differently, please let me know. I kind of forget what people do and don't know because I've been doing this for so long. So no question is too simple or too stupid. Please let me know. I might just go through now and let's have a look. I am going to bring up one more thing as well, which is I'll leave some notes. So don't worry, anything that I cover, I'll put some notes up on my Patreon. I am going to make an Obsidian file as well that will contain this information. Okay, so what we're going to start with is, sorry, 1 second. What we're going to start with is glucose metabolism. So this is kind of the easiest way. It's what I like to start with. It basically is universal, so it's a really good way of doing it. And what we're going to think is that basically these nutrients are just cycling in the environment all the time, right? Things get created and then broken down and then recreated continuously, and there's no beginning and an end. And you'll see this a lot in this. There's cycles, and the whole of Earth is in flux and is always in cycle. So the key concepts for this are that we take glucose and we turn it. So in fact, let's zoom in here. This is probably a bit easier. So what does this look like? Well, let's have a look here. So we'll start with this one. Oh, there we go. Okay, so if we have a look here, this This right in the center is glucose. We're going to ignore the pentose phosphate pathway for the moment. We're going to have a look down here, glycolysis and gluconeogenesis. Okay, this is what we're interested in. We're going to start with glucose, and yeah. Okay, we're going to start with glucose. We're not going to worry about this. This is from the liver for now. We're just going to worry about this central pathway. I'll actually bring something else up that explains this a little bit easier. Glucose gets broken, we'll talk about this in a moment. Glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate. And then this basically has a couple of different pathways it can take, but it basically ends in something called pyruvate. And in doing so, it releases ATP. This is, I'm going to bring up something that explains this a bit more simply. But basically, what is ATP? It's adenosine triphosphate, and we can think about it as the way that your body stores energy. So I'll show you what that looks like, and I'm also going to bring up a simplified glycolysis pathway. Glycolysis. So it's the word glycolysis. Lysing is to chop or break, and glyco for glucose. So to break down glucose. Okay. And how does it do this? Like, what are we talking about when we're talking about like energy and creation and stuff like this? So basically, what happens is, I've got a really good picture of ATP here that I'm going to bring across. Okay, so what do we mean? What is this? What's going on? When we're talking about energy, we're generally talking about something called high energy bonds. And you put a lot of energy into them, and when you break them, they release a lot of energy, which is really useful for us. And what are we using this energy to even do? Well, generally, we want to create, we want reactions to take place that are energetically unfavorable. And I'll show you what that means. So there's a bunch of, we're talking about anabolism and catabolism. So we always we can't create energy from nothing, and we always want to be moving in certain gradients. This, if I miss anything or you don't understand at this point, please let me know and I will explain. Basically, there is two kind of ways that things, I might just explain this quickly. Okay, so I'll bring you full screen. Okay, so basically let's have a think about this. If I want to say build a strand of DNA, I'm going to be linking molecules and atoms together. When I'm doing that, there's two things that can happen. There could be a high energy bond, and when they connect, a little bit of energy gets released, and then they reach a state of higher organization and lower free energy. So basically, it enables them to click together, and then they lose energy. So that's fine. And that process would be, if it's losing energy to create it, it is going to be catabolic, I would say. And if it could be anabolic as well, depending on the ways that it moves. But basically, it's losing energy to create something more complex. That's fine. Now, the other way is that you want to connect two things, but it's energetically unfavorable. So they actually need a little bit of energy to be able to link together. And you could think about it kind of like a carabiner with the gate, and you want to put your keys on them, right? If you put the keys on there, they're going to be more ordered. They're going to be linked together. But you have to put a little bit of energy in before you can open that gate. So the energy that's being used is ATP generally. And that ATP can be stored in all sorts of different ways. And we use this to modify molecules to create things that are energetically unfavorable. So there's a way that we could say, for example, so let's just cover that again. There's a way that requires energy. There's a way that releases energy. So there's maybe like two Lego bricks and we put them together and now they're more ordered. But when we do that, a little bit of heat gets released, for example. And there's a third option, which is you can change the state that things are in. So this is called isomerization, and an isomer has the same elements but arranged differently. So a good example of this might be a bunch of Lego bricks. If you have 10 Lego bricks, you can make a bunch of different things, say 3 different structures that are in different organizations. Maybe one's 10 high, maybe one's 5 wide and 2 high, and maybe one is 3 along, maybe 9 along and 1 on top. All of those would use the same elements, but they might all have energetically the same state. So we call this isomerization, and we're going to see this in glycolysis. So I'm going to bring you back now. So we've got these 3 ways. We can add things to each other, we can chop them up, and we can move the atoms around. Now, generally isomerization, it's really useful for us because generally it doesn't require more energy, but once we put them into a different state, we can do different things with them. So like the Lego blocks, it might be able to fit a different conformation. So by moving those Lego blocks around, you might make something that's more favorable. So maybe instead of having 3 prongs, you have 2 prongs, and you have another one that has 3 prongs, it might fit better. And that might enable us to do some different things. Okay, so with glycolysis, as a big overview, let's have a look. Okay, this is what we've got. We start with glucose up the top and we end with pyruvate, and we've got 2 chains. Now, if we have a look in a bit more detail, there's these side things going on. So the first reaction in glycolysis is to turn it into glucose 6-phosphate. So let's just have a look at what glucose actually looks like. Glucose. Okay. Glucose, here we go. So it's a sugar. What is a sugar? It's a carbohydrate. Carbohydrate is carbon, hydrogen, and oxygen. They're arranged in a circle, and they have numbers. So we can have a look at this. Let's have a look at this. What does this actually mean? Okay, basically at each point, at each corner or vertex, at each corner, there is an atom. So to look at it, it would look like this. The black ones are carbons, the red ones are oxygen, and the white ones are hydrogen. But it's basically carbons joined in a circle with some oxygen. It's got 1 substituted oxygen. Let's have a look. There's 1 substituted oxygen in there. So it's carbon, where do we number from? Here. Carbon, 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 oxygen. And the last carbon actually comes off here. We're not going to talk about too much why that is, but there's 6 carbons. There's 5 in a ring with an oxygen, and then one of them has an alcohol group, this OH coming off it. And let's just have a look at, let's have a look here. This is the one that I think might be easiest for us to understand. This is important because it shows the, there we go, it shows the numbering, okay? We're going to modify this molecule. So, I'm going to shrink this down a little bit. Okay, so we're going to be modifying this and breaking it up. At the end, the pyruvate is going to be chopping it in half. Okay, let's go back now to glycolysis. So we start with glucose, this molecule. Now, what was the first thing we're going to do? We're actually going to spend some energy. We're going to use a molecule of ATP. So that's this one here. So ATP is a modified amino acid, adenosine, S-I-N-E, adenosine, like amine is an amino acid. We can see that because it has the nitrogen means that it's a protein. It's got an amine group here. It's attached to another type of sugar. So this is a ribose. And then it's got, so adenosine triphosphate. And then it's got 3 phosphate groups. Now, each one of these phosphate groups between the P and the O has a high energy bond. So there's 3 high energy bonds stored in here, but we generally just use the last one and we convert it to adenosine diphosphate. So we take 1 off, it becomes 2 phosphate groups. Anyway, so basically here, this is our battery. We're going to take, the first step is to actually invest energy into this. So hexokinase, whenever we see the word kinase, we're talking about modifying a phosphate group. Hexokinase, and the reason for this hexo, we're turning it into glucose 6-phosphate. So let's just have a look at what this looks like. If we just exit here, glucose 6-phosphate. Okay. So we can think glucose 6-phosphate, we're going to be modifying something on the number 6 group. So if you remember, there was an alcohol group here. We've taken off the hydrogen and we've added a phosphate group from ATP. Now we've got glucose 6-phosphate. That required 1 ATP. Remember this because this will be important. We're investing energy in and later on we're going to get it back. But even though it makes quite a lot of energy, we have to invest a lot of energy too. So from glucose 6-phosphate, we have glucose phosphate isomerase. Isomerase, ASE is an enzyme. Whenever we see this ASE, it's going to be talking about an enzyme, which is a little machine that's going to modify the molecule. Generally, it will be, if it's an ASE, it will break it down, but it could also construct it. An isomerase will be shifting the same atoms around. It's going to reorganize the structure. And generally, we hope it's not going to take energy. And if we have a look on the side here, there is no energy investment. What does that mean, though? If there's no energy investment, it actually means this reaction can take place both ways. It can move forwards and backwards. This is really, really helpful because this means we can have regulation inside of our cells. I won't talk about that straight away. We're going to come back and we're going to talk about a very special case of this using fructose, which is related to fructose metabolism, which is why Americans are fat. Basically, it talks about the regulation of glycolysis inside the cell. Okay. Basically, so we've got this little bit here. It's going to move the phosphate. It's going to attach a phosphate group where the alcohol group was before. Okay, perfect. That's going to happen right here. That was an alcohol group, and now it's a phosphate group. Oh, sorry, I've got my screen on the opposite way. So if I'm pointing here, I'm actually pointing here. Okay, so we've got this happening here. We go to fructose 6-phosphate. So this is a change. Let's have a look. So fructose 6-phosphate. Let's have a look. Okay, so we've got glucose. We modified it to have a phosphate group. And then we're going to have fructose 6-phosphate. Okay. Fructose 6-phosphate. Okay. So what's going to be the difference between glucose and fructose? And we'll find out why we're doing this as we go down. I think it's easier just to go through it one enzyme at a time, what's actually happening. This is kind of the way I learned it. There might be a better way. Or, okay, let's have a look what's changed. Fructose 6-phosphate. This is quite a big change. Here we go. So we can have a look here. What happened? Okay. We had a 6-membered ring. 1, 2, 3, 4, 5. Okay, we've gone from a hexagon to a pentagon. The phosphate group has stayed where it is. It's on the 5 carbon. What actually happened was we've taken a carbon. Let's have a look. There's the oxygen. There's the oxygen. That's in the same space. So it goes carbon, oxygen. And then we've actually moved this number 2 carbon has come up to here. And the 1 carbon has jumped up. So there's a bond made between this oxygen and this carbon. That's what's happened. So it hasn't actually added or removed energy. It's just linked. Let's have a look. It's taken the bond between the oxygen and this carbon and moved it to this carbon. But everything else has stayed the same. So then we've got something called fructose 6-phosphate. So it's an isomerase that's able to do this. Okay, so now we've got this. Let's have a look what we can do. So we're here, phosphofructokinase. So it's going to work on fructose. It's going to be doing something with the phosphate. And in this case, we've got another investment of energy. So this will be the second ATP we've invested. We're taking an ATP and we're converting it to ADP to fructose 1,6 bisphosphate. It's going to add a phosphate group. So fructose 1,6, I'll just go here. I'll add another one. Fructose 1,6 bisphosphate. This should do. There we go. Fructose 1,6 bisphosphate. Okay, so this is going to be basically the same thing, except it's now going to have a phosphate group on the 1 and the 6. So if we have a look, we had it on the 6 before. We're now going to have another phosphate group coming off up here. Is that right? Let's have a look. Yeah. Okay. And it's going to be a very symmetric molecule now. There we go. Phosphorus 1,6 bisphosphate. Yep. So that's what it looks like. Let's have a look. Basically, it's exactly the same thing, except it's got a phosphate group on both the 1 and the 6. Okay, cool. This symmetrical structure is always really good because it means we can generally cut it in half and then do fun stuff with it. If you can get to this state, you know it's probably, there's a good chance it's going to be cut in half in some way. Now, it's not going to be cut perfectly in half, though, is the only thing, because of this oxygen. You can't cut an atom in half. So we're going to have an oxygen. Oh, there's another oxygen over here. It's going to look similar, but I don't know if it's going to make two exactly equal products. So let's have a look. That's right. Yeah. So here is where it splits up. This basically creates two slightly separate pathways. I'll see if I can just bring up a glycolysis process molecular. So I don't have to flip between these all the time. It should make it a little bit easier. Okay, so basically here, the initial phase where we invested the 2 ATPs is called the investment phase. So this requires energy to get started, to change it into something that's easier to break down. And then later on, we have we have this snipping in half section, and then we have the energy generation. Okay, so that's basically what this one's explaining here. Let's have a look. Is there one that has the molecules all the way down where we can see what this looks like? Okay, here we go. This is, let's have a look. Glycolysis. Okay, yep. So we've had glucose. It highlights a bond. Glucose 6-phosphate, fructose 6-phosphate, fructose 1,6 bisphosphate, this kind of symmetrical looking one. And then we have this step. So it is going to chop somewhere in the middle. Aldolase. So we're going to have a phosphate group on each side, a couple of carbons. Probably we started with 6 carbons, so we're going to have 3 carbons on either side, and the resultant molecule is going to have, let's have a look. So this is the enzyme that acts on it. We don't have to worry too much about that now, and this is the reaction we care about. We have this molecule, fructose 1,6 diphosphate, biphosphate. We're going to chop it. Where does it chop? There's an OH group here. Okay, I'm going to copy this and we're going to work out exactly where it chops it in paint. Okay, that's good. Let's paste this. Okay, let's have a look. I'm going to get a marker out and we're going to try in red work out whereabouts this is broken. So what I'm going to do is I'm actually going to highlight a group. Oh, that's too small. I might just resize this. Resize. I'm just going to make this 25%, just so it's a bit easier. And then view 100%. Okay, that's a bit easier. Okay, that makes sense. Increase my brush size. Let's get 12. Okay, that's better. Okay, so how do we work out where this is? We're just going to say that this phosphate group is this phosphate group, and we'll see if that works. We have phosphate, carbon, carbon. So 1 carbon, 2 carbon, oxygen. 1 carbon, 2 carbon, oxygen. Okay, that can work. 3 carbon, alcohol. 1 carbon, 2 carbon, 3 carbon, alcohol. Okay, so this is 1. But there's this oxygen here with a double bond. It only has a single bond here. What we're going to do is we're going to include it. So that's 1 molecule. And this is aldolase. So aldolase, let's have a look. Aldolase, did this require any energy to do this? No. So there's no new carbons being added. No new phosphates or anything being added. We're just chopping it. Once it's chopped, they can swap between each other, but it can't go backwards. So these should have the same atoms at the end. The other one is going to be, so that's going to be this one, dihydroacetone phosphate, and then this one is going to be the other. Here we go. Okay, there we go, and that's going to be this one. Now let's just count how many carbons each they have. We're going to say 1, 2, 3. So it's going to be C3. And then 1, 2, 3. This is C3. C3. 1P each. We can just write P. P. This won't be perfect, but this is just so we can count the atoms. Okay, pink. We're going to do oxygen. 1, 2, 3, 4, 5, 6. O6. 1, 2, 3, 4, 5, 6. O6. They should have the same, I think. And we don't worry about the hydrogens. So that's fine. Okay, yep, so they've both got the same amount. They're in a slightly different configuration. What is the different configuration? Are they mirrors of each other? No. And it has to do with, if we have a look here, there's actually a hint. One is acetone and one is aldehyde. And this is basically talking about functional groups. So what I'm going to do is I'm just going to erase all this. Dun, 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 dun. Okay, so what actually are these? These are... I forget the name of this. It's the special name for this branch of chemistry, but basically this talks about aldehydes and ketones. An aldehyde and a ketone basically are centered around this part. Okay? So this is a ketone. The reason we know it's a ketone is it has an O double bond to a C, and then it comes off like this with a C and a C. I think the way I remember this, a ketone, is like an open mouth singing. So this is the C coming out of the open mouth. And that's how I think a tone of C comes out of a ketone. It's an open mouth that goes into a C and then that spreads. And then an aldehyde is the opposite, or not the opposite, but the other one, which is this, which is an O bonded also to a C, but it's bonded to a C, but the other one is what's called R. It can be anything except a C. So in this case, it's actually this hydrogen here. So basically, one is an aldehyde and one is a ketone. We know that, so the tone and the aldehyde, the ketone and the aldehyde, we can just look at this functional group and say, oh, okay, it's that bit that's different. But energetically, they're probably almost the same. So they can move, well, they would have to be, because they can move backwards and forwards without requiring any assistance. So now we've got that out of the way. Okay, now we've split glucose in half. We've taken sugar, we rearranged it, we've split it in half. We put some energy in to do that, to rearrange it, attach some bits. Okay, now what do we have? We have, let's have a look. Let's have a look. Okay, oh, where are we? Let's shoot this down. Okay, so we're here, and we've got two separate pathways. Basically from here, we don't need to worry too much about it. What we're going to, I'm going to cover this quite quickly because it's actually like, from here, I think it's not actually that complicated. Basically, what we're trying to do on either side is we've rearranged the atoms, we've put them into a certain configuration, and now what we're going to do is two things. One of them is this first thing, which is basically we're going to rearrange the atoms on this pathway and do nothing on this way, but then the processes are going to mirror each other. This next reaction is generating NADH from NAD+. This is an energy gaining reaction. NAD is called a cofactor. We're just going to completely ignore this. It's not important now, but it is important later on. We'll actually come back to this. Then the next one, it chops off a phosphate. When it does this, we get one ATP back. We've got an ATP on either side. So let's have a look at this molecule. So yeah, basically we're going to get one energy out from either. So this above here is our break-even point. We have a dehydration reaction that happens, and then we get another one. So we've invested 2, and then we get 1, 2, 3, 4. So we double the amount of energy that we start with, okay? Well, it's hard for me to point because I'm on the wrong side. We double the amount of energy we started with. That's probably the take-home message here, and we're left with pyruvate. I'm going to show you quickly what this looks like at the end. Okay, so what's actually happening? We have glucose, we rearrange it, we have a 5-membered ring, we make it symmetrical, we chop that in half. Then here we generate we have 2 different molecules now, but they basically look the same, right? They have a phosphate on either side. Okay, so, okay. This next step, we add a phosphate group. This is what I was thinking, like, what, wait a minute. This is important. Here we go. Glycolysis. Phosphoglycerate kinase. We invest. So we've invested up here an ATP. This one, this NADH, NADH plus, this step here, we're actually going to, wait a minute. There's something missing. I'm getting something wrong because we need an extra phosphate group. I'm going to try and work out whereabouts that's going to happen. Let's have a look here. So when we cut this in half, we have 1 phosphate group. Here we also have 1 phosphate group. But here we have 2 phosphate groups. 1,3-PG. Let's have a look here. 1,3-PG. 1,3-PG. Okay, this step here. The enzyme acting on it, glyceraldehyde 3-phosphate dehydrogenase. Phosphate dehydrogenase. It's going to be taking hydrogen away from a phosphate group. We're going to have a look at this reaction here. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate dehydrogenase. Let's have a look at this. Let's have a look at this reaction. I think that there's something else happening here. Okay, if I've lost anyone, please let me know. Please stick a message in the chat. I think I can see a chat, but I don't think anyone's put any messages in there. Let's have a look what's going on here. Okay, so we've got this. This is this molecule with one phosphate group that we're starting with, and it ends with two. Okay, so there was something missing in this, which is when the NAD plus goes to NADH, it's grabbing the H from the phosphate group. So some free-floating phosphate group is going to be incorporated in. It's going to add this on. So magenta carbon, that the aldehyde group we were talking about, loses the hydrogen. Okay. And a phosphate group gets attached. So that is, let's have a look with paint. So this one here goes, and then another phosphate group, not from here, but just floating around, is going to pick it up. And this is good because we're generating molecules of ATP, which means we need P's to make it TP from ADP, and they can't come from nowhere, and they're going to come from this molecule. So we're getting towards the end now. We're on the home stretch. So this reaction is going to, the purpose of this next reaction, this one here, is to add a phosphate group to glyceraldehyde. Now that phosphate group is added, the next step chops it off. But when it does so, it creates a high energy bond. So we go back to having this. By putting the phosphate on, we're able to harvest the energy that was stored inside. Let's have a look at why that is. 1,3-bis phosphoglycerate. 1,3-bis phosphoglycerate? It is. They spelled it wrong. How fun is that? Let's have a look. So this is the reaction we're looking at. Right? Let's have a look. So we're going from 1,3-bis phosphoglycerate to 3-phosphoglycerate using phosphoglycerate kinase to 3-phosphoglycerate. Let's have a look. Let's have a look at how, now we might start to see the carbon skeleton is going to stay pretty much the same, I think, but we're going to be harvesting little bits of energy. So glyceraldehyde, this is what we're looking at. Glyceraldehyde, 3-phosphate dehydrogenase to glyceraldehyde, 3-phosphate dehydrogenase. Dehydrogenase. It's going to take, okay, so we added these together and then we end up with this. Okay, let's have a look. This doesn't actually show us what the molecule looks. Oh, that's it. Sorry. This isn't the right one. So we're looking at 1,3-bis phosphoglycerate to 3-phosphoglycerate. Okay, to 3-phosphoglycerate. Okay, here we go. So 1,3-bis phosphoglycerate. Yes, it's got the 2 phosphate groups. We have hydrolysis, so it's broken down. Hydrolysis, water breaking, pulls off an inorganic phosphorus, and it leaves this group here. Why is this important? Okay, I pick this straight away. This group here is highly reactive. Why? Let's zoom in a little bit and let's see what's actually going on here. Okay. This is what I learned in my entire degree, is how to look for things like this. I saw it straight away. By pulling off the phosphate, and that goes away, and creating this structure, this is unstable. Why is it unstable? Okay. It's because right here, you got the oxygen, and you got the oxygen. Oxygens are really what we call electronegative. When it's here, it's actually balanced out. When you have this structure though, there's going to be a competition for the electrons. So here what's happening is you have the carbon, and oh yeah, here we go. It actually explains it over here. Is that it's unbalanced. This double bond and this single bond and the hydrogen there are all fighting for the electrons. So basically, why is oxygen so electronegative? So this is quite important. What we're going to do is we're going to look at the orbitals of oxygen. If anyone has any questions, just chuck it in the chat. So the orbitals are where the electrons possibly exist. So the electrons and the protons balance each other out. So electrons, I think oxygen's about 6. So it has its first shell filled and it has almost its second shell filled, which is another S shell. So these are both around circles around 1s, 2s, yep, around the atom. So what's going on? We've got 1s, oh, it's got 1s, 2s, and then an almost full 2p shell. So I'll see if I can show you. Okay, here we go. So what's actually happening all the way down there? So you might have seen something that looks like this. We have this nucleus with protons and neutrons, and the neutrons keep the protons from pulling away from each other, because if you have two positive charges too close, they're going to repel. So we can stick a neutron in there to give some space between them, and allows them to exist, kind of like putting like a piece of cardboard between two magnets or something like that. It can stop it from pulling itself apart. And then we have some electrons, but they're not balanced to complete a full shell. We have a 1s, and then we have a 2s, and then we have a partially filled 2p. Okay, so what does this mean? What does this mean? Well, that's actually a bit of a bad way of explaining it. That's the pudding model, which Bohr used. We can actually think about it like this. This is much better. And they exist kind of somewhere in here. So right in the center, the 1s and the 2s, they're full. They're really stable. But then, to create a complete next shell, right, it actually needs full 2 electrons in the 2p1 X, 2p1Y, and 2p1Z shells. One of these shells is full, but 2 of the shells are not. So the green is actually balanced. They're sitting out there where they can kind of like interact with stuff. But the yellow and the pink here create really reactive groups. They really want just one extra electron, because they can actually only occupy either here or here. What actually happens is they're existing between them as possible electrons. We don't know until we measure it, but they can only be in one place. They're actually existing in a superposition now, but until they can exist in both places, so it could be here or here with equal probability, but both are full, it's going to be reactive. So these things are sitting out there trying to react with something, and oxygen and fluorine are the most electronegative of all. The reason for this is what's actually creating the pull is the protons. And because we don't have shells and shells and shells of electrons protecting the protons, kind of like the neutrons, it means that the protons are able to attract other electrons with the greatest amount of force. So this means oxygen and fluorine, because they don't have all these completed shells, they're the most electronegative. So whenever we see oxygen, we know something's going. Whenever we see oxygen with lone pairs or unfilled shells or things competing, we know a reaction is going to take place. We see that straight away. That's if you're ever in an exam, that's the first thing you're going to be looking for. Oxygens without complete shells, or in this case, we use this little delta, right? This little delta, let's have a look. This little delta is saying, well, it kind of has a complete shell, but not quite. And the reason for this is, remember, we've got 2 electrons that need to be filled. So the valence electrons and the carbon are all going to be fighting. So the carbon wants to make 4 bonds. It's actually only made 3 here, right? It's got one to the oxygen, one to the oxygen, and one to this other carbon. The oxygen wants to make two bonds. It's made one to the carbon, but what's actually happened is there's one electron being shared between all of this. You've got what's kind of a half bond. Half bond is very, very unstable. So we can use this energy though, this unstable energy that doesn't really know where it's going. We can actually stick that somewhere, and we can have a measure of this free energy here. Is that there's a more stable, more organized formation with lower free energy that can come out of this. And this is going to be the next step. So there's this free energy that's sitting here, ready to go into something else if we can somehow close these bonds better. And the ways that it's probably going to happen is, let's have a look. So there's an ionization, there's a hydrogen has flown away. We've got phosphoglycerate. What's the end of this? It is 3-phosphoglycerate. 3-phosphoglycerate. Let's have a look. So let's have a look at what it ends up. It probably ends up being something very much like this or this, but phosphoglycerate, resonance stabilization, yes. Okay, it's got a little negative. Yeah. Okay, so what's happening now is that actually the energy that we took away went to create an ATP. So we took some of that free energy away and we created an unstable structure here. Now what's going to happen is the next state will give it a more stable structure, which is 2-phosphoglycerate. So we're actually going to 2-phosphoglycerate. Let's have a think. 2-phosphoglycerate. 2-phosphoglycerate. Let's have a look what that looks like. So we've got this unstable structure, and we're going to create something stable with it, and it's going to be affecting that functional group with the 2 oxygens and a carbon that's unstable. So I think what we're going to do is we're probably going to get rid of an oxygen. Is that what we're going to do? 2-phosphoglycerate. Here we go. Let's have a look. Migration. Oh, no, we're going to move the phosphate around, and it's still going to stay unstable. So we're going to move the phosphate from the end of the chain up to here. And we're going to do that using an enzyme that can go either ways, and it requires a cofactor. So the reason you need magnesium is even though it doesn't participate in this reaction, it helps it happen. The magnesium doesn't jump on anywhere here, but allows a conformational change. So this delta G is the change in free energy. So this is an isomerization. We're just going to move the phosphate up. So we remember that doesn't really take much energy. So it can probably go either way. Yep. Basically, the free energy change is almost nothing. Enolase, we're going to have phosphophenoenol pyruvate. Phosphoenol pyruvate. Let's have a look. Phosphoenol pyruvate. Okay, let's have a look what that is. Phosphoenol. We don't want the enzyme. Phosphoenol pyruvate. Okay, so what's happening here? And we'll run, and then after, we're almost at the bottom now, and then we'll run through the entire process so we can understand what's happening at each step. Phosphoenol pyruvate. Now the final molecule will be pyruvate. Phosphoenol pyruvate looks like phosphoenol pyruvate, PEP. We actually want to see the step before this. In fact, we can go back to this one. We have it. Phosphoenol pyruvate. Where are we? I'm going to call this here we go. Phosphoenol pyruvate, PEP-ENO, is the enzyme that does this. So we move the phosphate here. Then we are, let's have a look here. 2PG, enolase. Let's just bring this up. Enolase. Okay. Let's have a look. enolase is going to catalyze the conversion of, okay, so it's a dehydration reaction. Anytime you see water coming off, it's called dehydration. But the reaction can happen in both ways, which means it's probably some sort of isomerization, because there's no real change in free energy, it looks like. What is happening is, oh, here we go. This is really cool. This shows you exactly how it does it. So what is happening is, oh, that's actually probably more complicated. Is this bit's the same? This is missing, so an H is going to come off. Oh, in fact, it's a dehydration, so two H's and an oxygen are going to come off. So this one's going to come off, and this. So let's just have a look what's going to happen there. Dehydration. Let's have a look. New one. Oh, shit. Okay, let's have a look here. Boom, boom, boom. Oh, oops, I actually copied image. Okay, let's have a look. What is happening here? Let's go through step by step. Okay, dehydration. So and whenever there's an option for things to come off, if water can come off, it almost always does. Like water's a very stable, happy molecule. It's always trying to come off. So we're gonna grab this hydrogen, and we're gonna grab this. We're gonna combine them to make that. So that gets chopped off. That stays the same. That stays the same. What changed? Okay. The C needs to bond 4 times. So that oxygen was actually bound to this carbon. So this carbon's missing a bond now. It needs 4 connections. At the moment, it's connected to 2 hydrogens and 1 carbon. And this carbon misses 1. So this bond here and this bond here actually becomes this bond here. So you'll see here there's a double bond. So now this is bonded to 2 hydrogens and twice to the carbon. So that's got 4. This is bound to the oxygen and the carbon and then twice. So that's got 4. So everyone's happy now. We've gotten rid of that. So basically the water came out, but the net energy didn't really change. It just swapped these 2 for another bond, and that was all fine. We won't worry too much about that. So it's not actually an isomerization, it's a dehydration. Not much energy change, that's fine. So now we have PEP. Okay, we have PEP. Phosphoenol pyruvate. This is starting to look very similar to the N molecule. So we have this structure, PEP. Now what's going to happen here is we are going to, okay, so what we did was, why did we bother doing this? What's the point? What is the point of doing this? Is that what we've done is going to enable this phosphate to be chopped off more easily by moving this away and by changing the strength of the bonds between this chain here. So basically what's happened now is that we've stuck something very, very far away. Where are we? CH2, that's here, that's here. We've got that bit up the top. Okay, and we've got this exposed phosphate group here. What we're going to do is we're going to chop it off and add it to an ADP using in an acidic environment. Super easy. What it's going to do is that's going to be energetically favorable reaction. We're going to take some of the free energy that's stored in the bond between the P and the O, and we're just going to move it across to an ADP and an ATP. To change engine ADP to an ATP. So basically what we've been doing is we've been trying to stick phosphate groups on, add some energy to them through different reactions, and then that stored energy is going to allow it to be transferred onto the ADP to create an ATP. We'll have a look at what the total things that we've done in this is at the end, and you'll see how this happens. So then we've created enol pyruvate, and then that gets moved to pyruvate. Let's see if there's any more ATP that's created. So we just did this reaction here, PEP. So PEP 2-pyruvate chops off the last of the phosphates, and that's the final reaction. Okay, so let's have a look. PEP 2-pyruvate. Let's have a look. This is the last one. And this is the final stage of glycolysis. So basically, we take a 3-carbon molecule with a phosphate on there, and we're going to chop that off and stick it back onto an ADP. What does it look like? Here it is, 3, so carbon, carbon, carbon. We're going to chop that phosphate off, and then we're going to be left with, we're going to have another one of these ATP molecules, which is the goal of the whole thing, and we're going to be stuck with pyruvate. Pyruvate is the end of glycolysis, and it has 3 carbons. That's probably the most important thing to remember. Let's just have a look at the type of bonds. When the phosphate gets chopped off, that oxygen then bonds back to the carbon to become a double bond. It actually gets shifted from here, which is good. And this change in energy is one of the things that we're interested in, because that energy actually gets stored over here. Okay, so now we've gone through the entire chain from glucose through to pyruvate. Let's have a look at what we did. I understand this is going to be very complex and probably go over everyone's heads, but let's just have a look at the broad process. We had glucose. We started with glucose up here. We used an ATP to put a phosphate on there. Remember, we're going to pull these back off. Okay, so we've invested one. We moved them around. We invested another one. And then we had fructose 1,6-bisphosphate. Okay, this is a 5-membered ring. We chopped that in half. And then, so we've invested 1, we've invested 2. We come down here, basically the same things. We move the atoms around. We add another phosphate group to each one. So we've now got 4 phosphate groups. We did this, so NAD and NADH are cofactors, but there's actually an energetic change that happens there. So we have 2 phosphates on each molecule now. Then we pull one of them off. We steal some energy. We come down, we move it around, we have dehydration, and we steal some more energy, and we're left with a little backbone. What we've actually been doing is stealing free energy that was inside the bonds between the carbons. That's the main thing that we've done. If you think about it, if we looked at the delta G between each stages of these reactions, the sum total was basically to pull it out and stick it on the phosphate bonds that enabled us to reconnect them to ADPs. Okay. So we took the glucose molecule, we added a phosphate, we chopped it, we added 2 phosphates, we chopped it in half, we added another 2 phosphates, then we sucked some of that energy out, gave it back to the phosphates to reconnect them. So we started with an investment of 2 and then a yield of 4. And then we're left with a carbon skeleton with 3 carbons and no phosphates. And instead we have 4 of the ATPs, which, as we've seen at the start, are ways that we can add energy to things. Okay, now I said it, that was very long and boring. I'm so sorry. What we're going to do now is we're going to talk a little bit about why is this important, like what this is done by every single living thing. Plants eat glucose. They have some different ways of creating some of the cofactors that allow for some of this energy generation to happen. Bacteria eat glucose, humans eat glucose, we all eat glucose. This process that we've just gone through is universal to all life forms as far as I'm aware. There might be some weird exceptions that prove the rule, but we'll just say everything has this process. This is central to life. Okay, so in here, there is some reactions that can happen both ways. What we're going to do now is we're just going to have a quick look at why this is important. Okay, this is reactions that happen both ways. Glycolysis, regulation. Because we might not want all of these processes to happen at once. We might want to regulate when this is happening. So say, for example, if you've got glucose, if you're a bacterial cell and you've got a bunch of glucose, but you don't want it all to be changed into ATP straight away. You want to eat it slowly across time, right? In fact, maybe you want to eat it when you need it because it might be useful in other ways. So let's just have a look here. The regulation of glycolysis. This is going to be really useful to us. Let's have a look. So where are these regulation points? This one's okay, but this... let's just have a quick overview of them. Actually, this is more complex. This is not good. This is better. Remember, I'm a molecular biologist, so I'm always going to default to these ones. Okay, so we start with glucose, and then we go into the start of glycolysis, where glucose to fructose 6-phosphate, that first reaction. And remember, this fructose 6-phosphate to fructose 1,6-bisphosphate, this reaction here requires ATP. Interestingly, if there is too much ATP in the cell, this is down-regulated. So ATP inhibits, an excess of ATP, wait, let me think about this, inhibits this reaction. If there's not much ATP, it would stimulate it to make more. So AMP encourages it, which is, we'll just say it's ADP and AMP are the same thing, it's monophosphate. Basically, the less energy that's available into the cell, the more highly regulated this reaction would be, and the more energy that's in a cell, the more down-regulated this reaction would be. So this allows the cell to work out whether it wants to eat glucose or not at this point. We won't go heavily into how that regulation takes place, just know that it does. The other one was at the end, PEP. These are two of the key regulatory steps. So PEP to pyruvate, that very, very last bit to harvest the final ATP out. Up until this point, it's actually a neutral amount of energy has been spent. When you think about it, that kind of makes sense. It's regulated the same way. So if there's an excess of energy in the cell, it doesn't complete that final step. It's not upregulated by AMP though, but it is upregulated by fructose 1,6-bisphosphate. So the product of this upregulates this. If you think about the entire thing, it basically means that the cell can balance its energy all the way across. We could work out why that is, but I've already been talking for over an hour. We're not going to go as to why fructose 1,6-bisphosphate would actually increase the end of it. But basically, it's talking about certain products, inputs and outputs, are going to regulate and feed back throughout it so it can create a smooth profile of ATP without using all of its energy in one go. Okay. So what is fructose? What is fructose? Fructose, right? So what is fructose and where does it enter? So we know we can eat glucose, but we can eat fructose as well, right? So if we eat high fructose corn syrup, right? Or fructose in general, which is in fruit, it's a type of sugar that enters the glycolytic glycolysis pathway at a different point. What does this look like? It looks like fructose 6-phosphate, right? It looks kind of like that. It's got that 5-membered ring. So they've got, wait, 1, 2, 3, 4, 5. It's got 5. It's got one less glucose. Okay, let's have a look at how it's metabolized. Fructose metabolism. So how's fructose metabolized? Well, it actually enters the glycolysis pathway. Let's have a look here. Fructose, it takes a bit of energy by fructokinase and it creates fructose 1-phosphate and then goes down here. But so it actually gets chopped up. Where does it get its bloody extra carbon from? Fructose is it 5? Is it 5? Let me just double check this. 1, 2, 3, 4, 5, 6. It's got 6 carbons. Okay. So it can enter the glycolysis pathway, fructose 6C, fructose 1-phosphate aldolase. So it's entering it as fructose 1-phosphate. Okay. Fructose metabolism, glycolysis regulation. Let's have a look here. And this is the final bit, and then we'll end for the day. Okay. Here we go. This will show you where it enters it. Let's have a look. Glucose enters here. So it enters in a slightly different way. I'm trying to find the one that will show you this the best. Glucose. Okay, yes. It goes down a different pathway, but it enters at the same bit as the aldolase. So if we have a look at what that looks like back here, it actually enters here on the pathway, the equivalent. It has a separate chain that looks kind of like this, but it enters here. Why is this important? Remember, it looks very similar to this, but it enters here. This is a regulatory step. So basically, if we have a look at the regulation, fructose 6-phosphate to fructose 1,6-phosphate. Okay. Fructose 6-phosphate to fructose 1,6-phosphate. Let's have a look. I'll bring this up. This step here is a regulatory step. Your body feels satisfied. Part of the feeling of fullness and the feedback from your cells is related to this step here. When you eat glucose, when it's enough, it down-regulates these pathways. When you eat fructose, it does not, because it doesn't get regulated until down here, which means you can eat a lot more fructose before you feel full. In fact, maybe you never quite feel full. So you can eat a lot more fructose than glucose. You will feel satisfied, a feeling of satiety, a lot earlier and with regulation up here, which means glucose is much better for you than fructose, because not because the end products are that different or anything like that, but one makes you feel full and satisfied. And that's because of this regulatory step here. Fructose skips this vital step, so you can just consume and consume and consume and consume supersized levels of fructose drink without ever feeling satisfied. Your body will say, yes, this is something I need, but it will never get to the point that it says, give me less. But with glucose, you do. And you can actually try this yourself. If you drink what's Mexican Coca-Cola in America compared to American Coca-Cola, it will actually, different amounts of it will make you feel full based around this part. Now, just to wrap up, so we've seen how this flows, we've seen some of the regulatory points and how this makes us feel satisfied or not satisfied. The final thing is at the end, we end up with pyruvate. And pyruvate enters oxidative phosphorylation. This is something that happens in animals and fungi, but it doesn't happen in bacteria. And this is how we can squeeze the final bits of energy inside there or between those bonds and pull out energy. And to give you to spoil the surprise, the carbon that you eat as glucose, it will break down that skeleton until the individual carbons come off. And the way they leave your body is as carbon dioxide. So every carbohydrate you eat that is processed and turned into energy by your body ultimately leaves as you breathing out carbon. So if you eat a Mars bar, any bit of it that's turned into energy and the carbon is not utilized in other parts of your body, most of that will actually leave your body via your breath. And if you think about how much a Mars bar weighs, or a piece of bread weighs, and how much your breath weighs, you'll see why exercise is so important for getting rid of that carbon. So all this carbon taken in is going to leave via your breath, and we're going to break pyruvate down into individual carbon atoms. And I'll be talking about that in the next lecture that I give at some point, which will be lecture 2 in this series. But that was glycolysis. I might do some follow-up stuff around on this if anyone's keen, maybe a second video just to address any questions. But if not, thank you for tuning in. I appreciate it, and make sure to follow my YouTube or my Patreon, and I'll be seeing